Well, good afternoon. I think we should all feel really privileged to be here in 2017 working in what surely must be the most exciting, dynamic, fast-moving area of the whole of science and the whole of medicine. I mean, personally myself, as a reformed organic chemist who saw the error of my ways, I very much feel that. So here we are, and if you can resist all the wonderful attractions of Copenhagen that you've just heard about and actually stick here, then, you know, we will have wonderful chances with presentations and posters and talks to hear a lot of the new developments in genetics. You'll be able to communicate your own novel findings. You'll be able to think about how this is going to affect your understanding of how humans work or what you can do for your patients. So quite properly, you're going to be entirely focused on the exciting present and the possibly rather frightening future. Nevertheless, for 30 minutes, I'm going to talk about the past. And the reason for that is because, as has already been said, we're celebrating the 50th anniversary of the European Society of Human Genetics, and we thought we can't let it go by without a little celebration. And as you know, the first meeting was held here in Copenhagen. Now, I mean, the story of how a society kept going, I think can be rather boring, can't it? I mean, you all know roughly what's involved when you set up a small society, and you all hope it'll work and it can flourish and prosper and so on. I would say, though, that the history of the ESHG is actually an exception to all of that. It's actually a much more strange and interesting story than your sort of standard history of how a society worked. So I'm going to take you through a bit of that. I've drawn very heavily in this on things from a number of colleagues, but particularly the work that Peter Harper has done, and he's kindly allowed me to draw a lot on his work. You really ought, if you're interested, to look at these two papers which are in the supplement to the European Journal, which those of you who are members can access online anyhow. You can also pick up paper copies at the Nature stand, and they assure me they've got loads of them. So those are really interesting reading for furthering things. And also Peter Harper's interviews with many geneticists, but particularly Mohr, who's already been mentioned, are worth talking about. So, how did it start? Well, it started actually in an experience that I think was familiar to many of us European geneticists, even long after 1967, was that the best place to meet European geneticists tended to be at meetings in the States. That was certainly my experience when I got involved in these things, which was quite a long time after 1967, I should say. And so it all started from a little discussion between a number of geneticists, but particularly Jim Rennick and Anthony Edwards, that this was happening. It really wasn't a very good idea. Perhaps we should think about forming a European society, the American Society having been formed in 1948 and going strong by then. So they resolved to put a suggestion at the 1966 International Congress of Human Genetics in Chicago, and so they put up a message inviting people, no email in those days of course. There were a number of people involved in this, but probably these are the ones who played the major parts in it, and the end result of this was that at that meeting in Chicago in 1966 they agreed that they would found a society, and they formed a committee to make this happen. And the chairman of that committee was Mohr, as already mentioned. And this has been documented by Jim Rennick and Anthony Edwards in a paper that was in the European Journal back in 1995. So this is the man who not only played an important role in the foundation of the European Society, but really was the European Society for the first 24 years of its existence, and this is why I say it was a slightly strange and curious story. It was not, under Jan Mohr, a standard orthodox scientific society. So there it was. It was formally born on the 15th of March 67, and it had the things you'd expect. It had a chairman, it had a board. The board was 20 people. They were drawn from across Europe, not in any sense elected or representatives of their country, just, I think, either members of that original little group who set it up or good chaps that they knew. And indeed, they were all good chaps. There is a singular lack of chapesses in this, as you will see. I think the only female in this entire list is the Russian cytogeneticist. So there it was. It had a board. It had a membership subscription. It had 174 members. It had a bulletin, which was a little short-lived, but still it had it. Much more interesting is what it didn't have. You would expect that any new society would have a set of officers doing various things. It would have some committees doing various things. And above all, it would have elections. The European Society of Human Genetics did not have any of those things. So this sounds possibly a bit more like North Korea than it does about a nice scientific society. So was Jan Mohr a Kim Il-mohr planning total domination? Well, he had his reasons. He had his vision, which was set out in an interview with Peter Harper in 2005. What had happened is that many years before that, 1956, he'd been associated with the department here in Copenhagen, Thor Kemp's department, and they had agreed to host the first International Congress of Human Genetics in 1956. And what Jan Mohr saw was that really for something like 2 years, all of the energies of the department were devoted to, first of all, being the ones who would set up the Congress, and then all the arrangements for getting that to happen, all the financial matters and finding funds to make it happen. And for something like 2 years, says Jan Mohr, really the scientific work of the Institute ground to a complete halt. And that obviously made a deep impression on him. And he thought, if we had a society that tries to do anything like that, we'll destroy human genetics in Europe. So we won't do that. He says there's a risk of undesirable findings. I love his congressional elephantiasis and politicking. Dear me, very nasty. So maybe that was part of the reason, he says. I emphasise the frugality and simplicity, even if the cost of that was that it was not very democratic. So he had a bulletin, but this didn't really need the sort of editorial functions that Gert-Jan van Olmen now provides, because there's no editing. Typescripts are reproduced photographically, and basically the bulletin carried the abstracts from the meetings of the journal, and actually I think it's fair to say nobody found it terribly compelling, and it all rather petered out after a few years, and the abstracts moved to the Clinical Genetics Journal, which also Jan Mohr had founded. Well, I'm not going to talk about the history of human genetics, I'm talking about the history of the society, but if we just think for a moment what sort of world the society was born into, of course, we understood by then that DNA gives RNA, gives protein stuff. The genetic code had been finally sorted out a few years previously. But there wasn't a lot you could do with DNA. I mean, you couldn't do PCR, of course. You also couldn't do cloning. There was no cloning in plasmids or E. coli or whatever. There was no sequencing. On the human genetics side, yes, there was well-established study of pedigrees, and the syndrome identification dysmorphology side, that was extremely active, going great guns, doing a lot. The biochemical genetics was quite well established and doing a lot. And you could also do linkage, assuming that is that you could find some genetic markers, which in those days was largely blood groups. And actually, Jan Mohr himself was the man who discovered the first human autosomal linkage, the Lutheran-Secreta linkage, some years previously. You could also look at chromosomes, and that had been going for about 10 years, but of course no banding, no fish or any of those techniques. If you read Peter Harper's second article, you'll find something extremely interesting that may surprise you if you believe the story of establishing the human chromosome number, the Chio and Levan story. Have a read at that. It's a little bit of an eye-opener. So there we are, first meeting in Copenhagen, and my number, which I think I got from Peter, is 95 participants rather than 70. And after that, there were subsequent meetings in different places. What tended to happen is that simply one person, perhaps from the board or perhaps one of their mates, was sort of either volunteered or arm-twisted to organise what they called a symposium rather than a formal meeting. And these things didn't cover the whole field of human genetics. They were on very specific topics. So you heard the first one was on human genetic polymorphism, and the ones on topics like meiosis and mosaicism and so on. So they were quite limited things. They were held normally in the organiser's university accommodation, which in those days he could probably get for nothing. They were expected to be self-financing. The registration fees were meant to cover the costs of running it, and everything was done on Jan Mohr's principle of simplicity and frugality. He said delegates don't want anything posh, suggesting they should probably be put up in student accommodation. And so it went on, and the various meetings over the years were held in different places in Europe, depending on where the people came. I very much wanted to be able to show you some photos of the people involved in those early meetings and, you know, a few formal pictures of the podium at those meetings. But actually, despite putting out calls to members, and thank you for those of you who did send in photos, but none of you managed to send in anything earlier than 1979. If any of you are still sitting on or think you might have access to pictures of pre-1979 meetings, we would very much like to see them, because we're thinking of setting up a sort of photo archive. So the first pictures that I've got are of the Southampton meeting in 1979, and even those are not of the meeting. We used to have an excursion for the meeting, and it's a very nice, decent excursion to Chichester Cathedral and on the boat to the Isle of Wight. I don't know who any of the people are. If anyone does know, please let us know. Bit of a change from the sort of stuff that we do now. That was Barcelona, or even worse. I won't tell you where that was, some of you will remember. So you had the meetings and you had the excursions, and so it went on, and I'm not going to take you through all of these, but from the Zurich meeting they had a trip out, I think, to the Rhine Falls at Schaffhausen, and this is the first photo in which we've actually been able to identify anybody. So Albert La Chapelle fortunately has a fairly distinctive profile. And so it goes on. Berlin 86, this was the first meeting I went to when the ESHG meeting was subsumed into the International Congress. And of course that was a very interesting meeting, because there we were in the conference centre, but this was before the wall came down, so there was some really interesting edgy tourism to be done. And carrying on, Corfu, just to show you that a few eminent persons came to these meetings. So it sort of pottered along in an unpretentious, low-key sort of way, but there were problems. And the problem was that as one went through the 80s, genetics was becoming more and more important, both in science, in medicine, and in public affairs. But the European Society of Human Genetics was becoming less and less important, because its meetings were just these little matey limited affairs with a very small number of participants. There wasn't really any terribly good reason why you should be a member of the society, because you didn't get any discount on the meeting fees, and anyway they were very modest, and the whole thing was sort of beginning to run down. And of course this probably first surfaced in the 1988 meeting, which was chaired by Peter Harper in Cardiff. And subsequently, at the next meeting in Groningen, it was agreed to form a committee. I mean, Peter had actually had a members' meeting. That was a rather unheard of democratic innovation. And from the discussions on that in 1989, they agreed to form a committee to just look at options to change things, make recommendations for new statutes. And eventually the new statutes were approved at the 1990 meeting in Corfu, where I've shown you some people. And that, apart from Peter's writing, also Giovanni Romeo and Eberhard Passager and Albert Lachapelle have prepared an article which hasn't appeared yet, but there will be a second ESHG supplement appearing later this year, and it's in that. So there we are. Now, Giovanni Romeo has pointed out that actually it's probably no coincidence that these things started happening in 88, 89, because of course 89 was an extremely significant revolutionary year in Europe, wasn't it? When everybody felt things, really could change, and let's have a bit of democracy, out with North Korean models. Giovanni also pointed out 88 was the first year that the European School of Genetic Medicine ran in Sestri Levante. And I think he's quite correctly said that that was pretty instrumental in forming a sort of community of European geneticists. So it's Giovanni and Victor Makuzik who jointly did it. It was modelled on the long-established Baja Symposium. So once again, we're bringing our fresh ideas over from the States. They had these workshops, which you can see were pretty intensive. I mean, look, there's a nice autorad. Some of you may remember trying to make sense of the splodges on them. I think that's probably Gert-Jan telling someone what it all means. Oh, my apologies, my apologies. So there was really this community of geneticists appearing, and so 1991 in Leuven was when the new order was established. Jean-Jacques Cassiman chaired the meeting. I think you'll all admire the understated elegance of that structure behind him, which was a sort of mascot for the meeting. I believe it represents 2 chromosomes rather than 2 Belgians caught in flagrante, but there it is. So the new statutes were approved. Many people were involved in this, but I think it's fair to say Peter Harper played an important role in this. And because this happened in Leuven, the newly renovated ESHG was formally registered in Belgium. Now, this has come back to bite us, because we're now moving forward to 2017 in a rather odd position. Our society is still formally registered in Belgium, so every year Jerome has to prepare tax returns for the Belgian authorities, even though we actually have no activities whatsoever in Belgium. But of course, all our administration for a number of years has been run from the Vienna Medical Academy, so all our administration is based in Austria, and we have some reserve funds to cover any possible disaster with conference and so on, and those reserves are for historic reasons held in a charitable foundation in the Netherlands. So we're split between 3 countries, and we're no longer the sort of little society which lives underneath the level of notice of the tax authorities. So Jerome is having to negotiate with tax authorities in 3 countries, plus whatever one hosts the conference. We try to live honestly. We don't wish to cheat, but it can be quite difficult to do it, and it's taking up a lot of energy. And we strongly feel that it's about time we did something about it. So we have the ambition to actually move everything to Vienna. The problem is exactly how to do that. I'm not going to spend time talking about that now, but if you want to come, those of you who are members, if you come to the membership meeting on Sunday evening, then Jerome and I will say a little bit more about how we think it might work and what's being planned. All I would say is that at the moment, nothing irreversible has been done. We're just exploring options and seeing how we can do it. You will also get a drink and some nibbles if you come to the meeting. So, the new order, we had an elected president who holds office for 1 year only, and Giovanni Romeo was our first president. And because they hold office for 1 year only, we had a president-elect, who was Margareta Mikkelsen. Here she is with Mette Warburg, sort of my tribute to the great Danish geneticists of the previous generation. It had a Secretary-General, Eberhard Passager, and that again had a turnover, so Jean-Jacques Cassiman took over, and then Peter Vanden, and then Helena Karinen, and then Gunnar Hauge, and now Karin Vritzel, who you saw on the stage. It had a program committee headed by Charles Bausch, which after a bit I took over and then was able to hand the poisoned chalice on to Han Brunner, and who in turn had Brynhilde Wirth and now Joris Feldman managing it. We had the journal, and the journal was founded in '93, and Giovanni did this, and he edited it for the first 2 years. So we're not only celebrating 50 years of the Society, we're celebrating 25 years of the journal as well. And then Gert-Jan van Ommen took over in 1995 and has been with us ever since then, to our very, very great relief, and appears willing to carry on. So here he is with Peter Vanden, our very active Secretary-General from the 90s, looking, I thought, a bit Mephistophelian. And we have committees, and perhaps the most important of these, which has already been mentioned, is the Public and Professional Policy Committee, which was set up originally through work from Ségolène Aimé, who's looking appropriately serious here, and has been run for the past 10 years by Martina Cornell, and who is going to hand over to, got my senior moment, Francesca Forzano, in the next year or so. And that, the slogan there is responsible translation. So being responsible about translating the many, many scientific achievements into actually ethical and practical service to patients. And they do all sorts of things, but Olaf has already mentioned a number of these reports. They issue formal guidelines which go through extensive consultation, and we have these various statements and press releases that we put out. And so they've done exactly what Jan Mohr was frightened of, haven't they? We've got congressional elephantiasis. Here we are, 3,000 plus of us. He'd have had kittens. And politicking, and the PPPC is actually quite a big committee. There's about 15 people on it, and they're pretty active. So this is a lot of active politicking. But then, you know, if you don't talk about stuff, then people who don't know about it are going to talk about it. I mean, nobody's going to stop public debate on issues of genetics and its applications, and no one's going to stop governments getting advised or hectored on genetic issues. It's probably quite a good idea if people who actually know what they're talking about do it, rather than people who don't. So it is, I think, very important that we should do these things. So there you are, we've got that committee. I'm not going to spend a long time running through all the other things we do, but we have the courses and fellowships. Currently, Han Brunner chairs the Education Committee, and we spend a lot of money on courses and fellowships. And of course, the focus of that has been the European School of Genetic Medicine, currently in Bertinoro. Just a few weeks ago, we had the 30th iteration of it. Many of you will have been to it. Any of you who have not been to it and who are at the fairly early stages of specialist training in either clinical or laboratory genetics, I would very strongly recommend you to find out about it and consider going, because I think if you talk to people who have gone, our feedback is that they've all found it very positive and very useful. And we have other sponsored courses as well. And we have relationships with lots of other bodies, particularly the European National Human Genetics Societies, where we have joint memberships with quite a few of them. We offer a range of conference fellowships. Some of you here will be on fellowships offered by your national society, but funded by the ESHG. We have meetings of presidents of the societies. We have cordial relations with the American Society, which you can see in a number of ways, but we have the Building Bridges session on Monday morning, which is a jointly organized session. the International Federation of Human Genetics Societies, where the current president is our own Helena Karinen, other clinical specialties, as Olaf has mentioned. MPAG is a rather curious, I was going to say organisation, but I'm not sure if it is an organisation. It's a recurrent meeting. It started out actually back in 1988, I believe, in Groningen in the Netherlands, but for quite a few years past, on even-numbered years, so not this year, but last year and next year, they have a formal meeting in conjunction with the ESHG. There are discussions going on about how to move forward on that, and lots of other things. Let's not go on it. But of course, the main thing is the annual conference. I mean, that is undoubtedly our major activity, and that depends crucially on Jerome. This is actually Jerome and his team at the first Congress that he organised in Vienna in 2001. And it's proved to be a wonderful relationship. I was actually involved when I was head of the SPC before then with some other professional conference organisers, and one of the more interesting episodes that happened there was that just as I was checking through the final proofs of the abstract book, which of course in those days was a thick book of all the abstracts and you know, deadlines looming hard, I suddenly realised that they'd very tidily rearranged every author list of every single abstract into alphabetical order. So unless you were Mr. Abrahamson, you weren't going to get any first authorships, and unless you were Mr. Spigniewski, you weren't going to get any last authorships. So I can tell you that was a few sleepless times. So it's worked wonderfully with Jerome, and in fact it's gone so well that in, I think it's 2007, we asked him to become our executive officer, so he's the only actual paid employee of the ESHG, and it's so wonderful that I've given him a halo. And the other person that one should mention in connection with the conference is Janti de Roos of Roos International, who actually since 1993, her firm have been responsible for everything that involves outside companies. So all of the exhibitions, all the company satellites, all the bits of sponsorship on your lanyards and so on, that's all Janti's work. And without those two, the conference simply would not work. So there we are. That's a sort of quick history of how we got to be where we are. And here we are, and let's hope this is going to be as good a conference as I think it should be. So I will stop there, and thank you very much.
Thank you, Andrew, for this very inspiring talk with the black British humor, what we are used to it in the board.
Right,
so I think it was for everybody inspiring, and many aspects of this, what we didn't know, at least our younger, older generation. And I think we should sort of remain the history for the next years somehow, and we have a discussion in the board how we can remain all this information what are gathered over the next year, that for instance the slides and the people who are on the slides we clearly can define for the next years, for the next century. Thanks, and I hand over to Joris Veldman.
All right, it works. So it's a personal pleasure to introduce Han Brunner. I have to say I was a little bit worried when I read Peter Harper's piece in the European Journal, in which it turned out that Han Brunner was already interviewed, which means that he's part of the history of human genetics. But clearly, when we read his lengthy abstract, we see that he's very much part of the presence of human genetics, and it's a great pleasure to have him here. Han Brunner.
Thanks, Joris. Should I do this? Yep. So here we are. This is the present. I'm here to discuss it. I accepted the invitation to speak about the present in human genetics and then reflected and decided this was really I only accepted out of extreme vanity to be on this stage and tell you about now is ridiculous, because now is this meeting. It is you. It is what happens in the next 3 days. So I found this a little daunting, and I decided to just put in bits and pieces that I think have been significant over the last few years. I have one disclosure. I'm going to sound very enthusiastic about next-generation sequencing for diagnostics. Some of that is driven by the fact that we sell these tests to some other countries. Our departments have limited income from this. I have no further disclosures except that I was going to wear my nice suit, but that's not going to happen today. Now, this is a quote, and it's a quote many of us use, and apparently Sidney Brenner denies he actually said it, or at least he said it quite this way. But it's a quote that I think many of us would agree with, and it is: progress in science depends on new techniques, new discoveries and new ideas, and probably in that order. And of course that's the bit that is sort of upsetting if you adhere to this more romantic vision that the great idea comes first and then you design the experiment and so on. But that, I think, is very true for human genetics over the past few years. And we've had lots of innovations and lots of new technologies that gave us new insights. And I'm going to mention a few people along the way. Most of these have had our society's award of excellence in science, and this is the ESHG award. And here is Peter Lichter, who got our ESHG award in 2012 because of his important role in developing fish and arrays in the field of cancer genetics, but also with applications to all of human genetics. And it was arrays, really, that gave us CNVs and genomic disorders. Completely novel concept just 10 years ago, and now such an important part of what we do. And Evan Eichler and Jim Lupsky from America especially drove this development and drove this application of this new technology to understand human development and disease. Now, just over 10 years ago at ESHG in Amsterdam, we had Sidney Brenner speak. And he spoke in his inimitable way, and somewhere halfway through his talk, he explained about this new machine that he'd seen and heard of in Cambridge locally that was being built. It was all very secret. It was not yet on the market at all. And I think all of us in the audience thought, is this real? Because what he was talking about was the first Selecta next-generation sequencing machine that, together with 454 sequencing and a few others, really started this era of next-generation sequencing and human genetics. And I distinctly remember coming away thinking, this really changes everything, and it did. Because we got whole genomes. And first, of course, it was the Watson genome done on a 454 sequencing machine. Craig Venter also had his genome done. That was done by conventional Sanger sequencing and not by whole genome sequencing, but it was done by shotgun sequencing. Then some human geneticists started sequencing themselves or their friends, and then they turned into sequencing the more interesting people. And there were quite a few celebrities, including movie stars and Desmond Tutu, I think the Nobel Prize winner from South Africa, was sequenced. Now that sequencing has really changed everything. And it's become so normal in just a few years. And I think the most impactful application at this moment in time really is non-invasive prenatal testing. I point here to Rosa Chu and especially Dennis Lowe from Hong Kong, who really drove this with many other people. And now we see non-invasive prenatal testing being applied as national programs, and often to all pregnancies in a country. And in the Netherlands, we just started doing this for all women, starting from April 1st. And it turns out about 50% of pregnant women in our country will take the test, which adds up to something like 80 to 90,000 a year. So these are very, very large numbers. When you do that, you start seeing other things. This is from a paper that Joris Vermeersch did 2 years ago from Leuven. What they found, and a few other groups have found the same thing, is that this test is so sensitive because it detects such small amounts of DNA that you can actually see malignant cells' DNA. Because malignancies, of course, those cells often are destroyed, and then there's a bit of free DNA that ends up in the bloodstream. And the NIPT test detects it as something very, very strange. So the test says there's a very awkward and strange chromosomal abnormality we don't really know. And then if you look more carefully, you might find that the woman has a cancer. So this is the concept also, of course, of liquid biopsies. That hasn't quite happened yet, but is about to happen, we all think. Joris, since I have him on screen here, also discovered using arrays something that I think none of us had ever thought was possible. And that is that the human embryo in the first few stages of division is a mosaic. It's a mosaic with a lot of abnormal cells and very few normal cells. And how there could be 3,000 of you here just beats me. Because if you look at the early embryos that you once were, that is impossible. What did you do? You probably lost those abnormal cells by selection, and only the very good cells survived. So this is an insight about mutation, variation, arising spontaneously, and then very, very strong selection. And I think this is one of the real messages that we've learned over the last few years, that selection is so important. There used to be papers by people who said, oh, human genetics, selection is gone. There's no more selection. We're a finished product. Well, not so. It's happening all the time. Many groups have started using this in the diagnostic arena. We've been very happy with this for the last 6 years, even though, as you can see, these machines are now very, very antiquated, and you would not even find them in a museum, I think. And that's just 3 years ago. It's unbelievable. This is a very strong test. Exome sequencing is very good, and it's very good across the board. It gives you reliable and diagnoses for a large percentage. Well, not a large percentage, maybe between 20 and 60%, depending on the disease. But as we and others have shown, that's much higher than if you ask doctors to guess what the gene is going to be and then order a Sanger test. So the average yield using this kind of genome-wide, maybe exome sequencing or genome sequencing as a diagnostic test is about 3 times as good as what doctors get for many, many of these conditions. And this, of course, means that by now exomes we do more exomes locally at Nijmegen for developmental disorders than we do Sanger sequencing for these particular patients. And that, I think, is a trend that's going to continue. So there's this enormous convergence of genomic technologies, starting with karyotype and array and FISH and Sanger and MLPA, and it all sort of merges into NGS. And I think that's, very few of us would now doubt that this is where it's going. There are obviously other technologies, and people are inventing new things as well, but NGS does seem to be here to stay and to sort of become the main engine driving human genetics forward. One thing, and this is the first time I ever dared to put this on a slide. I think we've now identified collectively most of the monogenic disease genes. And the reason for saying that is from a paper by Kim Boycott, who is chairing the diagnostic committee of ERDIRC. And the ERDIRC organization objectives by the year 2020 are to deliver 200 new therapies for rare diseases and the means to diagnose most rare diseases. And ERDIRC is basically just a framework for a lot of national scientific organizations from around the globe. And in a paper that just came out recently, Kim and her colleagues have tabulated this. And this is the new genes that were found and identified in any given year, starting 2010 to 2015, which is the last where the numbers are complete. And as you can see, there's a curve here, with the high point being 2013, when there was this enormous bonanza of new disease genes found by next-generation sequencing. But since that time, the curve is going down again, slowly, but it is going down. And the percentage of new conditions where a gene is found, where the gene is a known gene, is increasing. So we've probably found most, at least most, of the monogenic disease gene. Not all of them, there will still be new ones, but they will become rarer and rarer as we go along. Because we now can diagnose these rare diseases, of course, people are becoming more aware. It used to be, I remember, see, I should probably have done the history talk. Well, I couldn't because you did a much better job of it. But a lot of my sentences now start with I remember, which probably means that I'm beginning to be part of history. But anyway, I distinctly remember that many of the surgeons and oncologists in my hospital did not really believe that breast cancer was inherited. And that's 20 years ago. And only after BRCA1 and BRCA2 were identified that they sort of grudgingly admit that, yes, perhaps some of the time breast cancer could be genetic. So it's only when you see things that you realize they actually exist. And this is very true for rare diseases generally. So now, because we can now diagnose them and show they exist, people are beginning to take an interest. And this is really important because rare disease diagnosis is still a very slow process. And this is from a survey done in the UK in 2010. It shows that most patients with rare diseases, if they are diagnosed at all, see between 5 and 10 different doctors before they finally find one who actually understands and knows their disease. So there's a real need for experts on these rare diseases, because there are so many of them, to work together in Europe and combine all the knowledge. And that is happening. And it's happening largely because, or at least in part, because of the activities of Sigala and Aimé, who started Orphanet many years ago, but has also been very influential in getting the European Reference Network accepted and then put to work by the European Commission. And there's now over 20 European reference networks for rare diseases. Most of these are based on genetic diseases, and they will combine knowledge, infrastructure, and new novel therapies and so on across Europe, hopefully in the next few years. If we connect knowledge, if we connect the knowledge we have collectively as specialists in genetics and in rare diseases, then of course we need to also collect and combine the patients. And that's happening through this kind of initiative where different databases that contain descriptions of rare patients and their genomic variants are combined through a matchmaker exchange. And this is a really important way in which we can find, if you have a patient with an ultra-rare disease, and they often ask, do you know another patient? And you have to say no. But then you put them in the database, and perhaps someone in Turkey, or someone in Malta, and someone in Finland will say, well, I've actually seen exactly the same thing. And this is a really important development. At the same time, of course, that we create these resources, beautiful as they are, the patients are solving the problem themselves, because a lot of them now find each other by Facebook. And we've had a lot of experience with that, where the patient just go on the internet, start a Facebook page with the name of the gene in it, and all the other mothers come around and say, oh, I'm so glad I found you. And this obviously is a way forward, and it's a way of thinking that we need to embrace. So we've identified most monogenic disease genes now, I think. But this started a long time ago, and Lena Peltonen got the ESHG award in 1997 for studying the molecular basis of these Finnish heritage diseases, originally described by Reijo Norio. And one reason why they are there is because the Finns really are on the outer limit of Europe, as Lena used to say. And they are at this limit genetically, and they are quite a distinct genetic group. So they have their own diseases, and these are mostly recessive diseases. This idea of studying specific populations, and now studying it at scale, also came to This man who got the ESHG award in 2009, Kari Stefansson, he was in Reykjavik. I think he was in Boston when he had this idea. I think the idea came to him partly because he wanted to go back to Iceland, and he wanted to do genetics. So then, of course, he added it all up and founded Decode Genetics, which now existed for almost 25 years. And I got this quote from their website, and they say they have collected genotypic and medical data for more than half the population, the adult Icelandic population. And I've seen quotes by Kari that say that they can now impute, if they wanted to, because of all the information they have on the genealogy and on SNP markers and so on, they could impute risk genes for basically any person living on the island and born on the island, which is an interesting concept, and they are planning to take this forward and do basically populations genetics, but also translating this into population genetic care. They did a paper, this is Augie Kong, also working with Stefansson, where they used their database and their genomic information on a lot of people from Iceland to calculate the rate of de novo mutation, which is about 70 to 80. Other groups have found the same thing, and actually this is very, very close to what people had predicted many years earlier in the absence of any data. Strangely enough, this is one of the few predictions that turned out to be correct. All predictions about the number of genes that we have turned out to be completely wrong, with most people fancying 100,000 or so at least, but as we now know it's just over 20,000. And they also, this graph from that paper from 2011 shows that the mutation rate increases with paternal age, and it does also increase with maternal age, but much less. And that means that there is the risk of the older father. And the risk of the older father translates into a number of diseases that often arise through de novo mutations, such as achondroplasia, skeletal dysplasia. And the gene for that was found by two groups. One was in the United States, and the other was Arnold Munich's group at Necker. And Arnold, because of all the work they did on, not just on identifying disease genes, but also on identifying and developing new therapies, including therapies for achondroplasia, he got the ESHG award in 2008. Now, achondroplasia is interesting because it's much too frequent. Because basically it's just a single mutation. And why do we all know people with achondroplasia? It doesn't add up. And it was Andrew Wilkie who recognized that this was wrong, that there were far too many of these patients, and then proceeded to investigate what was going on. And it's not mutation rate. The mutation rate for this particular gene and this nucleotide isn't actually that elevated. What is elevated, what elevates it, is a process that happens in the testes. And Anne Gorielli and Andrew Wilkie together worked on this and showed that if you look in the, certainly in an older men's testes, you'll find these small clones that have these kinds of mutations. They're activating mutations, and they somehow activate sufficiently that these mutant sperm cells increase relative to other sperm cells. You can find them. The idea of de novo mutation is broadly applicable. We applied it to intellectual disability, and after looking around, found that at least 60% of intellectual disability in non-consanguineous populations is through de novo mutation, which is quite a high percentage. It's much, of course, it's much more skewed towards recessive if you have consanguineous populations, but in the Netherlands, where we have very little consanguinity, it's mainly de novo mutation, both structural variants and nucleotide variants. And of course, we cannot prevent de novo mutations from happening. They're essentially random. And that's another thing we learned. So, presumably then, once the technologies have advanced beyond chromosomes, and once the technology is there to actually call and then interpret single nucleotide variants arising de novo, I would certainly envision that we'll have not just NIPT for chromosome abnormalities, but NIPT also for de novo single nucleotide gene changes. And the reason for that is that there's actually the combined incidence of severe intellectual disability through these de novo mutations is probably higher than that of Down syndrome, and the severity is also, it's more severe. Complex disease genetics has progressed by genotyping technology, LD maps, imputation, statistical innovation, and certainly by very, very large sample sizes. And it was the HapMap project that gave us these LD blocks, where if you know one SNP in the block, you can predict what the other SNPs are like. And that, together with very large sample sizes, has given us a lot of GWAS hits. Now, people used to show this, and when they added all the hits up, they didn't explain much of the variance. And then people had this slide that said, what we're probably missing is something here in not just the Mendelian disease variants, we can see those, and but not the common disease variants, we can see those as well, but probably the low frequency disease variants in the middle. But that actually turns out to be not true, and they're probably very rare. What is happening is something very different, because height is still very strongly genetic. But then people did a GWAS of over 250,000 people, published in Nature Genetics in 2014, and found 9,000 genetic variants which explain 29% of the variance. And I made this rather unkind slide that said if you want to make a prediction of how tall your children will be, measure your height and do not measure your genes. And that was true at that point, but I was wrong, because actually this brilliant man, Peter Vischer, who's in Australia, and other people, of course, there's never just one person, but he invented a number of statistical innovations and computational innovations to look at SNPs in a better way and impute them. And that, from that, this really important paper, I think from 2 years ago, basically says that if you now do the calculations again, and you take all the little bits and pieces that are there in the GWAS data, then there actually is no missing heritability, and all the heritability is actually explained by these very, very small pieces, by this very large number of common SNPs. So this then goes towards solving a problem, another problem that existed 100 years ago at the time of Johansen. And this was Mendelian versus blending inheritance. And blending inheritance was basically statistics. And this comes from Galton and Pearson of the chi-square, who said, you have to do statistics. And this, of course, we still have a community today, and that's the complex disease genetics, who say, you need to measure it and then calculate it. And then on the other side, there are people who believe in individual genes, and the monogenic people. And they fought for a while about who was right. And of course, as in most of these debates, usually both sides have a point, and both are right. So is it polygenic or Mendelian? If you go back to height, most of it is polygenic, but certainly if you look at another isolate, or a semi-isolated island, genetically and historically, Sardinia, then you find that the variants there have drifted up. The common variants have drifted up to give you shorter height, but there's also some almost monogenic variants that have a real impact on height, including one in the growth hormone receptor gene that reduces height in heterozygotes by about 4 centimeters, which is not insignificant. So it really is a combination of things. Autism, there was a lot of debate about autism. Is it monogenic? Is it polygenic? Both sides showing their data, saying, I have data to show it's monogenic, and the other side saying, I have data to show that it's polygenic. And of course, it's both. And some of it depends on IQ. If you have very low IQ and autism, then you're much more likely to have a de novo severe loss of function new mutation. If you have a very high IQ and autism, you're much more likely to have a familial and an inherited variance of the multifactorial GWAS type. And this is a very recent paper again from Mark Daly's group in the US, and I included this quote that says, we find that polygenic variation contributes additively to risk, even in ASD cases, so autism cases, who carry a strongly acting de novo variant. So common disease often arises by a mix of common and rare variants. NGS informs cancer sequencing informs cancer treatment. Mike Stratton got the ESHG award in 2014 for leading the International Cancer Genome Consortium, among other things, and for identifying BRCA2. And this, I just wanted to show you this. This is, of course, the Philadelphia chromosome, which Janet Rowley showed to be a translocation. And I wanted to include this picture because she's very lovely here being given this medal. And I was also very pleased that it was this particular president giving her that medal. 10 years on, a group from Erasmus University in Rotterdam showed that this translocation actually creates a fusion gene between BCR and ABL. And in 2001, imatinib was developed by GlaxoSmithKline, I believe, to treat CML. And this, of course, has been a paradigm, but it's been a long time before we had multiple others. There's a few others now, but I think this is now, the pace is going up, and this is the most exciting development, I think, now. This is Steve Jackson from Cambridge showing that he's not done pipetting for a very long time, I think. But it is now known that 20% of ovarian cancers have BRCA mutations somatically, and that most of these, but the majority of these patients actually have one germline mutation. So it's a combination of things. It's a combination of a BRCA germline mutation and a BRCA somatic mutations, meaning that if you sequence the cancer, you're basically doing genetic testing on the woman. And I think that's an interesting paradigm, and we're going to see a lot more of that. The bonus, of course, is that you can then administer olaparib, this drug that blocks the alternative DNA repair pathway from BRCA1 and 2 called PARP. John Byrne always points out that PARP inhibitors were developed in Newcastle, so I need to mention that too. So there's a convergence of germline and somatic mutations with relevance for treatment. The next step probably is going to be organoids. Hans Clevers in Utrecht, other people are developing these. And the ideal picture there, I think, is that if you could make an organoid quickly from a cancer, you could then try different drugs on that organoid to see which one was most effective. Very similar to testing bacteria for their resistance or sensitivity to antibiotics. And it will be a while before that can be done on an industrial scale, but who knows, it might happen soon. Treatment is difficult, but it's happening. This is from the French, Paris, Necker again, Alain Fischer and his team, Marina Cavazzano Calvo and Salima Assing Bey Albina, who developed gene replacements, one of the first gene replacement therapies that was developed in the world, in this case for SCID, or the bubble children. And we're all looking for exon-skipping drug therapies, and this gentleman has done a lot of work on that. This is him with his girlfriend, probably about 50 years ago, just under 50 years ago, when life was a lot wilder, and that's Gert-Jan van Ommen, who got our award in 2011. I just got that from the internet. It's not like I tricked him. There's a convergence of phenotypes that I think is really interesting. If you look at the genes involved in inflammatory diseases like Crohn's disease, ulcerative colitis, or even type 1 diabetes, MS, and so on, celiac disease, there's quite a bit of overlap. And it seems that we're hitting the same genes or the same variants a lot of the time for different phenotypes. For neuropsychiatric diseases, again, the same GWAS hits, and this is in terms of co-heritabilities between bipolar disease and manic depressive disorder, perhaps not, or major depressive disorder, perhaps not surprising, but then also between schizophrenia and major depressive disorder and so on. So there's a lot of genes and variants that do not increase or decrease risk for a single disease, but actually for a range of diseases. And the same thing is true for de novo mutations. This is from a review from David Goldstein in 2014 that shows that genes actually do not respect our diagnostic boundaries. The nosology we use to describe our patients is not accurately reflected in the genetic findings. So we may end by having to rewrite some of our diagnostic truths. This is very recent. One paper from Nature Neuroscience last year that showed that FOXO3 variation, common variation in FOXO3 is linked to brain size in the normal population. Just this month, there is a paper in Nature Genetics of a genome-wide association meta-analysis that defines new loci in genes influencing human intelligence, and their top hit, again, is FOXO3. And presumably, these are the same variants, although I did not go into the supplementary to find out about that. So again, although we can see that is not illogical that there would be a connection between brain volume and intelligence, this is something that the genetics is now telling us. It's not something that was done as part of when the experiment was designed. So I'll take 2 more minutes and talk about the future. So this is also, I think this is the most exciting development at this point, that genome analysis is going functional. There's ENCODE, the Encyclopedia of DNA Elements, which basically is a lot of RNA and chip sequencing. And that, coupled with transcriptomics of single cells, makes us transit from defining the traits, the inherited genetic factors, to the state. So we're becoming traiteurs rather than members of the State Department, which is, I think, is an advance. It's a way forward, but it's a new way of looking at it, because now we stop looking at DNA and we start looking at RNA. And a lot of the understanding comes from thinking about functional domains, and you will hear from Edith Hurd, who was this year's ESHG awardee. I think she was the first woman to get the award for 20 years, so the committee needs to think about that very carefully. Well done. Find some more women, I would say, because they're there. Yeah, I think they got the message, that's all right. So, Edith Hurd and Bing Ren in the US really defined these topological domains that are about concerted gene expression and their boundaries and so on. And this is fascinating, and it's being applied to human genetic disease, and Stefan Mundlos got the ESHG award 2 years ago for this work on patterning abnormalities of the links, together with the next speaker, Axel Wiesel, so I'll not tread on his toes and let him talk about this soon. So how will this time be remembered? I think this is the time when we've actually identified human monogenic disease genes. It's not over, but if you want a career, probably look somewhere else. There are still many more GWAS hits to find. If you have ever larger sample sizes, we will find ever smaller pieces. They will be informative biologically. Now, there's a lot of interest to do that, but you need to be part of a very big consortium. We still, at the course, we still have students who are starting a complex disease genetics experiment or a PhD on 100 patients. Do not do that. Join a consortium. Prenatal non-invasive diagnosis may soon be universal. Monogenic and polygenic make up a continuum. It sounds very self-evident. There probably is no missing heritability, and that we now have most of the genetic pieces, we need to understand the functional puzzle. Cancer treatment, hopefully, will soon be by functional readouts and giving us much better choices of treatments. So I'm going to thank a few people from the department at Nijmegen, especially Joris Veldman, who unfortunately has decided to join Newcastle University, so he's gone. Thank you very much.